Yeah, thank you so much for the invitation to come speak here. So I'll be telling you about some of my PhD work and future work on machine learning for determining protein structure and dynamics. So I just started my group a couple months ago at Princeton's computer science department. There's kind of two main scientific directions that I'm interested in. The first is kind of pushing the envelope for determining dynamic protein structures from single particle cryo-EM data. So this is continuing my PhD work, which was done joint with Tristan Bepler, who's now at NYSBC, and my two PhD co-advisors, Joey Davis and Bonnie Berger. And so the task there is, given a data set of unlabeled 2D images, reconstruct a distribution of 3D structures. And I've been lucky to kind of have wonderful collaborators that we've been kind of iterating on this method. So Adam Lair, we worked together on CryoDragon 2, which I will cover briefly in today's talk, and then also recently working with Axel Levy and others at Stanford on CryoFire. And Vineet has been a research software engineer who has been fantastic in helping me kind of take over maintenance of CryoDragon as a software package. The second direction, which I won't be covering today, is in situ structural biology with a cryo-EM and machine learning based approaches. So that is something that I'm very excited about, and I'd be happy to talk to others about offline. So I wanted to motivate this line of work, even though we're at a math institute, in both the computer science and the biology perspectives. So from the computer science perspective, we've had kind of major breakthroughs in machine learning algorithms applied to protein structure prediction. So we have kind of AlphaFold and RosettaFold, which have taken this input protein sequence and been able to train this large statistical machine to predict the 3D structure. And that's kind of exhibited by their 92.4 GDT score at CAS14. And even kind of, and I think one thing that's very interesting from that machine learning standpoint is that AlphaFold has provided these architectural building blocks that have really opened up new areas. So just a week ago or 2 weeks ago, the folks at FAIR have connected AlphaFold architectures with large-scale protein language models to then predict the structures of 617 million metagenomic proteins. So I think this is a very exciting time for ML and structural biology. But what are the new frontiers? So I'm particularly interested in cryo-EM because these machine learning systems are ultimately systems trained from data, and cryo-EM is this modality that can produce new data. So for these problems like complexes, protein-protein complexes, what are their structures, what are the dynamics of these protein complexes and their interactions, I think kind of cryo-EM and cryo-ET are the route moving forward to really develop next generation machine learning systems. Of course, the biological motivation is just as interesting. That's why this field is so cool. All essential biological processes are carried out by proteins and protein complexes. And so if we can understand their structures, the goal of structural biology is that we can better understand their functions. And so obviously we can understand these fundamental molecules in all essential biological processes and also develop kind of new therapeutics to intervene in medical and health applications as well as in nanotechnology and biotechnology applications. This is last month's PDB 101 Molecule of the Month. Unfortunately, so these images are just static pictures. They're really beautiful. They show kind of the different shapes and sizes of these protein machines. But what's missing is that these complexes are actually very dynamic. And unfortunately, techniques to study molecular motions are rather limited. So we have NMR, which is limited to really small proteins, about 100 amino acids in length or shorter. We have what I like to call avant-garde crystallography-based approaches like EFX and XFL crystallography. However, it still requires sample crystallization, and you can really see some of the mechanical deformations, but this is quite kind of early days. My previous background was in molecular dynamics simulations. So what we see here on the right is an MD simulation of the SARS-CoV-2 spike protein, and while this gives you a very kind of precise in silico microscope at what might be happening in these systems, this is ultimately just a simulation, and so you'll still need experimental data to validate what's going on in terms of the dynamics of these molecules, which is why I've been very excited for the opportunity for cryo-EM at uncovering molecular motions directly from experimental data. So I won't go over the overall cryo-EM image processing pipeline. I assume we're all very familiar here. There's micrograph preprocessing. The area that I've been focused on is this 2D to 3D reconstruction problem. And then finally, given a sufficiently high resolution 3D density map, we can fit in an atomic model. So just zooming in a little bit more on single particle cryo-EM image formation. we have a purified solution of our molecule of interest that's flash frozen in a very thin layer of vitreous ice and then imaged with a transmission electron microscope. And we can model this process as the integral projection of our volume along this imaging axis. I don't have my pointer, but because these molecules are tumbling randomly in solution before being flash frozen, they're captured from some unknown orientation. And so we have, okay, giving up on the pointer, but we can model each image as a 2D function from xy to the cryo-EM density at the location, and it's a rotation of the volume along that imaging axis z, and then shifting by the 2D translation corresponding to imperfect centering of our molecule within the image, and then convolution by the point spread function of the microscope, and then finally additive noise, modeled as additive noise. So that's the cryo-EM forward model. The goal of reconstruction is to solve the inverse problem. So given a data set of experimentally determined cryo-EM images, a typical number being in the hundreds of thousands or even millions, can we reconstruct the 3D cryo-EM density within those images? So there are a number of challenges associated with this task. The unknown pose, or the orientation of each of the molecules within the ice. So I described that previously. The very high amount of noise in these images. You have to take into account image grading filters like the CTF, and then discretization of the measurements. Then the challenge that I'm particularly interested in is this so-called heterogeneity problem, which is where we break the assumption that each of these images captures an identical static 3D structure. Before I talk about the heterogeneity problem, just one more piece of background information, which is the Fourier-Slice theorem. This helps us relate the 2D projections to the 3D structures in this imaging modality. And the Fourier-Slice theorem states that the Fourier transform of a 2D projection of a volume is a central slice out of the 3D Fourier transform of the volume, and that orientation of the slice is perpendicular to the projection direction. So this kind of suggests a way of reconstruction. If we have a data set of images and we know their orientations or we know their poses, we can take the FFTs of those images, insert those as central slices in the corresponding orientation of those particles, build up our representation of our volume in 3D Fourier space, then take the inverse FFT to get back to our reference volume. So that's in fact how traditional kind of homogeneous reconstruction algorithms work. You can write down kind of the statistical framework for the data generation process and then try to estimate the volume or the model that best explains our captured images. And so in practice, this proceeds under this EM, or expectation maximization framework, where in one step we estimate, given some initial estimate of our volume, we can align all of our images to that volume. And then in the second step, we can take those alignments and then re-estimate the volume. So that's this kind of iterative coordinate ascent procedure. And there's many state-of-the-art software packages that implement kind of this traditional homogeneous reconstruction, Reliant and CryoSpark being two main software packages in use. So that's for the homogeneous problem. The heterogeneity problem is actually one of the major opportunities in cryo-EM. So each image here contains a unique molecule, which might be in a different conformational state. And so unlike in crystallography, where we've experimentally rigidified each copy of our molecule into a crystalline lattice, in cryo-EM, we actually get the experimental data of the potential kind of conformations of our molecule of interest. You can imagine how this opportunity is really a problem on the reconstruction side. So standard approaches with dealing with heterogeneity include kind of discarding as much of it as possible, maybe even experimentally rigidifying your system. And then the workhorse that we've talked about earlier today is 3D classification-based approaches, where we model image formation as originating from a discrete mixture model of K independent structures. And so a typical cryo-EM processing workflow looks something like this. This is a figure found in kind of the supplement of like most cryo-EM papers, where at the top we have around millions, we have like a data set of millions of picked particles. And by the end of this quite ad hoc, manually driven filtering process, we've kind of eliminated, or in this case, eliminated maybe 90% of the images, and we get two structures at high resolution. So the identification and analysis of heterogeneity, especially continuous forms, is an open problem in cryo-EM. So in this discrete 3D classification approach, you kind of need discrete initializations, and it might work well if you only have a few independent structures in your sample, but you don't know this a priori. So we would like to kind of develop more advanced forms of modeling image formation and conformational heterogeneity to be able to reconstruct continuous kind of complex distributions of structures. So the method that we developed is called CryoDragon. It stands for Deep Reconstructing Generative Networks. And here the task is 3D reconstruction of a continuous distribution of structures directly from unlabeled 2D images. And the main kind of hypothesis starting out was that there's been this like crazy development in large-scale machine learning, or basically neural network-based modeling techniques that can really model complex distributions using deep neural network architectures. So the idea was, can we adapt some of these approaches to heterogeneous cryo-EM reconstruction? So CryoDragon frames heterogeneous reconstruction as learning a deep generative model of density maps. And one thing that's been particularly exciting is that this architecture that I'll be talking about has shown broad applicability in other domains of computer vision. So CryoDragon at a glance consists of kind of these 3 different aspects. The first is our model for the volume. So we develop coordinate-based neural networks to directly approximate the 3D structure. The overall architecture for CryoDragon is this Fourier space image encoder volume decoder architecture that's based on the VAE. And finally, the way we do inference of this model is with this exact inference or search procedure for the unknown poses, and then variational inference in the style of the VAE for our latent variable describing heterogeneity. So the main idea in CryoDragon is how we model structures. And so instead of representing the structure as discrete points on a 3-dimensional lattice or in this 3D voxel array, we instead use a neural network or a fully connected multi-layer perceptron or MLP to directly approximate the function from 3D Cartesian coordinates to the cryo-EM density at that location. So CryoDragon structures are parameterized using this neural model instead of a voxel array. So the goal of reconstruction in the traditional case is to fit the value of our cryo-EM density at each of these voxels. In CryoDragon, we want to learn the kind of parameters, the weights and biases of our MLP to best approximate this 3D function. There's an additional detail here, which is that instead of directly inputting our 3D Cartesian coordinates, we featureize them using a fixed series of sinusoids of varying frequency. So this is a positional encoding function, and it was inspired from the transformer literature. And this is kind of the exact architecture that has been shown to be very useful for other tasks in kind of traditional computer vision, like novel view synthesis. So if you're interested in that, please check out NERF. And there's even been kind of popular science articles written about NERF. And so that's been very exciting to see. Just to give a brief vignette on kind of why this kind of sinusoidal featurization was important. Here's a toy example where we're using an MLP just to learn, or really to memorize, a single image. So we can think of an image as a mapping, a continuous mapping from XY coordinates corresponding to the pixels to the RGB values at each of those locations. If we're trying to memorize this particular image, this is the ground truth. Without the sinusoidal featurization, the neural network failed to learn the high resolution details, or kind of the high frequency details. With the sinusoidal encoding, then we can recover kind of all the fine-grained details in the image. So this was kind of a key development in CryoDragon that enabled us to learn high-resolution cryo-EM density maps. And if you're interested in checking or learning more about this, the authors of NERF have follow-up work that explore kind of why there is this spectral bias in fully connected neural networks. So why are we doing this? The main reason is to model heterogeneity. So in the kind of traditional 3D classification or multi-class reconstruction approach, we have K separate voxel arrays. And so again, there's this issue where you have to manually kind of pre-specify K and their initializations, which can definitely bias the results of the reconstruction. And typically you're limited to around 10 volumes, although I think later versions of CryoSpark say you can reconstruct hundreds of volumes in 3D classification, which is quite exciting. In CryoDragon, we can simply extend the input dimension of our neural network to include an additional latent variable vector. So what does this mean? So this latent variable vector is just this kind of generic latent variable that we can use to encode additional degrees of freedom for our molecule of interest. So if Z were one-dimensional, you can think of this as position along some reaction coordinate. If Z were kind of higher dimensional, which it typically is, we can think of this as encoding position on some conformational landscape. And so we can simply, with this kind of neural-based model, we can simply very kind of easily extend this neural representation with this conditional latent variable model. This encodes some n-dimensional continuous distribution over structures. And so that's the CryoDragon generative model. The question is, how do we learn such a model from our single particle cryo-EM image data, which is very noisy and looks like that. So our overall architecture is this Fourier domain image encoder volume decoder architecture based on the VAE. So given an input image, our encoder maps this to some location or some distribution in latent space. So that is the latent code, Z. Our volume decoder is this coordinate-based representation or coordinate-based neural network that I just described, which takes in our latent code and 3D positionally encoded 3D coordinates, and then predicts the value of our volume at that location in latent space and in kind of positional space. So given an input image, we can reconstruct, because of the Fourier slice theorem, each of these images corresponds to a slice out of our 3D model. And so we can get the coordinates of that 2D slice and then reconstruct the image pixel by pixel, compare the reconstructed slice with the input image, compute the loss, and then use that loss to update kind of the weights of the encoder and decoder neural networks. So one nice thing is that this coordinate-based architecture really nicely enforces this geometric consistency between the 2D views. We can apply kind of these pose orientations and translations as explicit operations on our input coordinates instead of having to model that directly in our neural network-based architecture. So that was assuming, and I'll talk about kind of how we do pose inference later, but just to summarize the training regimes, given an imaging data set, we jointly train these two neural networks to learn the encoding and decoding of some n-dimensional continuous latent space describing the data set's heterogeneity. Our objective is the standard VAE objective, and there's two training regimes. The ab initio case, where we're estimating poses on the fly, and I'll talk a little bit about that later. And then heterogeneous reconstruction using previously estimated poses. So this is possible where when there's not when the system isn't highly heterogeneous, and you can do a consensus reconstruction on maybe a relatively rigid part of the complex. So that's how we train the model. How do we actually use it? So at test time, we have these two neural networks. We have an encoder neural network that we can use to predict some latent code given each of our images. This is this lower dimensional representation, and maybe so we can directly visualize this, and maybe this tells us something interesting about our data set already. But we also have a decoder neural network that can generate volumes given different values of our latent variable. So we can view representative samples from these different locations in latent space. We have this continuous model, so we can, given two endpoints of interest, do an interpolation in latent space, generate the associated volumes corresponding to each of those locations, and view this continuous trajectory of volumes. And then if you don't like the neural representation, you can always kind of check that we've learned consistent reconstruction by taking a set of particles around that location in latent space and then performing a traditional homogeneous reconstruction using traditional tools. And in practice, this latent representation is particularly good at detecting outliers, and so it can also be used to filter the data set for various out of distribution or kind of heterogeneity that we don't want to model. A little bit about CryoDragon usage and workflow. CryoDragon, the compute requirements are standard for a single particle cryo-EM reconstruction software, so you'll need GPUs. And the actual code is implemented in PyTorch. In terms of the input data requirements, you'll need extracted single particle images, ideally high quality pose estimates, where secondary structure features are well resolved, and accurate CTF parameters. And then in terms of the software pipeline, hopefully it's relatively easy to use. So it's like a couple of series of commands that's shown on the left. there's a very extensive tutorial for how to run CryoDragon and interpret its outputs. Recently, I think actually today, the Nature Protocols paper was officially in press. And then there's been a lot of kind of enhancements and integrations that I've been very excited about, like various integrations with Chimera X and other interactive rendering tools. And then a lot of the support has been done by Vineet, who's a research software engineer at Princeton, who previously worked on cryo-EM with Amit. So he kind of came in already knowing a lot of the details of cryo-EM, which was fantastic. A couple of new developments in the latest release, although we're basically at version 2.0 right now. But one is how we interpret the distribution of results, which is this automated conformational landscape analysis tool, which will kind of cluster your states and give you discrete structures, which is kind of what structural biologists really want at the end of the day, and then kind of assess the numbers of each of these discrete structures. And the second is the fact that since there's been a lot of developments on the kind of computer vision side of things with the NeRF-based models, we've been able to take some of the developments there, bring them back into the CryoDragon representation, and modify the architecture for a higher resolution modeling and faster convergence. So that's been pretty exciting. So that's a quick overview of the motivation for CryoDragon and cryo-EM reconstruction and an overview of how CryoDragon works. Next, I want to talk about how we initially validated this approach on synthetic data. So starting out, I didn't really know what was a good benchmark data set for methods for modeling continuous heterogeneity. So we started with this hypothetical atomic model and then simulated a continuous degree of freedom. So I rotated one of the dihedral angles that connected the purple with the orange and blue part of this complex, and then generated 100 atomic models along this degree of freedom and simulated the cryo-EM image formation process. So the goal starting out was, can we reconstruct this continuum of conformations? Jeffrey, what's up?
Do you think that, so you sampled 100 models, and how close do we need to get to continuity where we can have every image sort of be a different point along this trajectory? Did you see any artifacts?
There were no, to my knowledge, artifacts from the actual discretization of this continuous reaction coordinate, but we could definitely sample this even finer if we wanted to, but just as for convenience sake, we just stopped at 100. And I'll show you the results, right? So these are the results. So you can tell me if maybe, I don't know, no, I don't think so. I don't think there's any kind of
Why isn't the ground truth stepwise thing?
Or I'm just ordering it along 50,000 images. So the ground truth technically is each 1,000 image chunk is a separate model, but for the sake of this plot, I'm just showing all 50,000 embeddings. So on the x-axis is the ground truth kind of ground truth reaction coordinate along that dihedral angle, right? And then the y-axis is the predicted latent embedding for each of the images. So we see this nice correlation between our latent embedding, which is this one-dimensional latent variable model, and the ground truth reaction coordinate. And then we can also generate structures from this latent representation. So here I'm showing 10 structures at these 10 points along Z, ranging from roughly -10 to 10. And here's a movie. And in comparison to CryoSpark, so that's shown here on the bottom row. Again, this is a couple of years ago and kind of the first work. So we were using very small images, and so these are relatively low resolution volumes. But with multi-class reconstruction, with 3 classes, we swept kind of k equals 1 through 10. There's one structure which resembles kind of the ground truth structure in gray. The other 2, at low isosurface, you can see that there's various alignment artifacts. We've also just lost all the heterogeneity in the data set. So here's another kind of data set we created, which has a dihedral angle that's rotated 360 degrees. So that's the circular motion of the orange part of the complex. And ab initio CryoDragon, here with a 10-dimensional latent variable model, we can recover this circular motion in the structures. And then What I'm showing there is the latent embeddings for the data set where the color is the ground truth reaction coordinates. So we have this nice kind of rainbow circle. And again, the multi-class reconstruction approach, there's one structure in gray that kind of resembles the ground truth, but the other two have weird alignment artifacts. And we've also lost all the underlying heterogeneity in the data set. So there's a couple of additional data sets with more complex latent structure. The linear 2D data set has two parts of the protein complex that are moving kind of independently. The circular 1D data set I've already shared. There's a mixture of 10 discrete classes of this kind of hypothetical protein complex. And we also quantify that CryoDragon can reconstruct kind of the distribution of structures. That's the nice thing about the synthetic data set. There's one additional detail, which is that we were playing around with kind of, instead of single particle reconstruction, encoding two images after doing a tilt series, and this definitely helps with representation learning of the latent space. I think that's what the images there are shown on the top row. The next kind of direction was really evaluating this on real data. So there's a couple of vignettes that I wanted to share. The first being applying CryoDragon to learn homogeneous structures and then to uncover residual heterogeneity in these data sets that are originally homogeneous. So the first data set is this Plasmodium falciparum ADS ribosome, or Empire 10028. We originally just trained the CryoDragon decoder with no latent variable input on the images from this data set. And so the neural representation is here on the left. The voxel-based representation, I believe, from CryoSpark is here on the right. We can kind of recover the high-resolution details, although it is slightly lower resolution, maybe due to the box size of this particular, the large box size of this data set. And then playing around with some of the model, just like architecture settings, to develop kind of best practices. We find that the representation capacity, or kind of the resolution of the models the resolution of the structures that we can model are limited, obviously, by the architecture, so the number of layers and number of kind of the width of the architecture, as well as the Fourier featurization or the positional encoding function, and then in the case of heterogeneity, what the dimension of the latent variable is. And so there was all these kind of just empirical tests to explore what's a reasonable set of parameters for CryoDragon. And of course, on a practical note, you have to kind of consider the inverse trade-off between the architecture size and the training speed. We picked this data set since it's also been analyzed with other techniques, so multi-body analysis, which models heterogeneity in terms of B rigid bodies. CryoSpark 3D VA fits this linear subspace model, and in these methods you can see that there's motion of the small subunit of this ribosome, and there's rotating motion. And so this is what we get with CryoDragon. I'm showing 20 sampled structures here on the right. From these different 20 points in the latent space shown here on the left. Now I have the mouse. Okay. One of these structures is from this outlying cluster here, which corresponds to this rotated state. But in addition to this large-scale rotation, we also see lots of other heterogeneous elements in the large and the small subunit. And so just showing that in a little bit more detail, we have this rotation. The mouse is gone. The concomitant kind of motion of this helix and the appearance and disappearance of this protein and RNA helices. On a small complex, so that was one question that people always ask is, how does this work on smaller protein complexes? This one is the RAG1, RAG2 complex, which is 10x, it's an order of magnitude smaller than the Plasmodium falciparum ADS ribosome, and we can still recover all the high-resolution details. And when we train the heterogeneous version of CryoDragon, we have the stable core, which is relatively homogeneous, but then we see all these DNA and protein elements that are kind of appearing and disappearing, or kind of folding and then denaturing below this stable core. So those were two data sets that were originally published as kind of homogeneous. The next test was applying CryoDragon on the assembling ribosome data set. So this particular data set studies the modular assembly of the large bacterial large ribosomal subunit. And so this was done by my advisor Joey during his postdoctoral work. The original data set has 130,000 cryo-EM images containing a mixture of assembly intermediates, which through kind of many rounds of hierarchical classification were split into 4 major states, B, C, D, and E, and then 13 minor states after this hierarchical approach. This is what we get when we train CryoDragon on this particular data set. So we trained a 10-dimensional latent variable model, and I'm plotting the embeddings of the images with PCA on the left, and then with UMAP, which is this nonlinear kind of dimension reduction technique on the right. We can already see that there are clusters in the latent representation. But because we have this 3D classification previously done, we can pretty easily validate that these clusters in the latent representation align well with the published 3D classes. So what I'm showing on the left is each image colored by the class assignment from 3D classification. And so the 4 major classes, B, C, and D, and E, are captured as those 4 kind of clusters. So that's a nice test, or kind of nice validation. And then we can also reconstruct structures given this latent representation. So what I'm showing here are structures reconstructed at the centers of each of these clusters. B is the least mature form of the large ribosomal subunit, so it's just kind of the back of this particle. C, we've built in the base. In D, in red, we have the central protuberance, and then in E, in purple, we have, this is the most mature form that has most of the elements of the large ribosomal subunit. We're not limited to just those 4 structures, so that's the beauty of this approach relative to 3D classification. Class A is actually an impurity during sample preparation, which is the 70S ribosome. And so this is mixed in. There's a couple thousand images that were mixed into this data set, and those get separated out very nicely as this tiny blue cluster. And then when we generate the structure at this location in latent space, we get the 70S ribosome. I'm also showing a couple different samples from class E, or this purple cluster, at the centers of the minor assembly states. So E1 is least mature, E2, we have this helix on the left, or kind of RNA stalk on the left. E3, we have the shoulder on the right. And E4, we've added this, docked this helix in. E5 is the most mature form of the class E ribosome, large ribosomal subunit. One thing that was very cool was that there was this strip of particles that were colored purple, but next to the green cluster. So what does this mean? This means those images were originally kind of clustered into class E, but when we generated the structure from this region latent space, we got a structure of the ribosome that resembled class C, which is kind of this less mature form that only has the base, but it had the helix docked, which was previously only associated with the class E, or most mature form of the ribosome. And when we take those particles, or the 1,000 images, and then do a homogeneous refinement, we get the same structure with this helix docked. So this was a nice validation and discovery of a new assembly state from the data set that was just missed to the kind of manually driven 3D classification. The last vignette is applying CryoDragon on a data set with continuous heterogeneity, or continuous motions, which was the original motivation starting out. So this particular data set has the structure, contains the structure of the precatalytic spliceosome. The original publication by Plashka, Lin, and Nagai published a static structure, or kind of a single composite map, that was pieced together from focused refinement of the different regions of this complex. This particular data set was also analyzed multi-body refinement, and there's this large motion of the top, or the SF3B and helicase subcomplexes relative to the core. Analyzed with CryoSpark 3D-VA, we also see this large-scale motion, but because of the linear subspace model, the motion kind of appears or manifests as this appearing and disappearing artifact. And this is what we get when we train CryoDragon on this particular data set. So what I'm showing here are structures generated along the first and soon the second principal components of our latent embeddings. And so along this first PC, we see the same kind of large-scale variability of the SF3B and helicase subcomplexes. on long PC2, they're kind of moving in opposition. One caveat that I wanted to mention, especially for users, is that these interpolations can produce non-physical motions, especially when your data set is not supported along this interpolation path. So for example, if you had kind of clusters like compositional heterogeneity, if you draw the continuous interpolation between the two, that'll definitely be non-physical. So this motivated this graph traversal algorithm. So here what we're doing is we're building the KNN graph of all of our latent embeddings and then doing kind of a random walk along this graph, generating structures at the visited nodes to generate what I think of as a plausible trajectory of this precatalytic spliceosome in motion. And so obviously I'm not making any claims on this is the actual trajectory, but I think this is a really nice way of exploring the learned distribution and just really seeing kind of the full distribution of structures learned by the deep generative model. Wow. Okay. So the last thing is a note on interactive, or how we want to analyze these distributions moving forward. So these two vignettes I've already talked about were on previously kind of analyzed data sets. So we had this nice way of comparing against the other methods. But as this tool has been now used in more like kind of in the wild, in real application settings, there's this question of how do we analyze the result of this deep neural network model. So we have kind of a latent space, we can generate structures. The approach taken originally is to provide as many interactive tools as possible to just explore your data set. So I created a couple of Jupyter notebooks to interactively visualize your latent space embeddings, plot them with different kind of, by different other metrics like your CTF defocus or your pose values, but also interactively visualize new images. So very relevant when you have crazy junk artifacts in your data, and interactively generate new volumes, though not as interactively as kind of the new Chimera X integrations. And so that's in fact very useful for many data sets that are not kind of the final filtered ones that are deposited on Empire. And so we find a lot of data sets that have non-structural imaging variability, and this actually impacts optimization of the model quite significantly. And so in this data set, like, you can't even see it here, but there's like a small little streak of particles at the very top where those like numbers are, and the images look like that. So that's clearly some like ice tearing artifacts or something along those lines. Here's another example where there are grayscale or kind of like kind of shifting artifacts on the cluster on the right. In this particular data set, without removing these outliers, the model really focused on trying to model these crazy kind of things on the side. And so you had to actually just go through a round of filtering your data set before, here's another example, which I forgot to take out. But you had to do that before we were able to recover the underlying structural heterogeneity. So that was, in fact, this particular data set, which is a SARS-CoV-2 spike protein. Through 3D classification, they found an RBD receptor binding domain down, or open and closed state. And after removing the junk particles, this was a very kind of didactic data set experience for learning how to train these models. We were able to recover the open and close kind of transition, which is very exciting. And just for some like data set and training details, this had 276,000 particles, took approximately 10 hours to train on 4 GPUs, not counting the filtering and the learning process. Recently, we've made some work towards automating this kind of analysis, right? It's really good to kind of interactively explore your data set and really become very intimate with all the quirks of it, but it would be very like what you really want is an automated approach and to be more kind of reproducible and quantitative with what you're doing. So just as an example, this was a data set, Su Fei, who's a postdoc in Bob Sauer's lab, where they were studying ClipXP. She trained a CryoDragon model. She got this particular latent space, very beautiful, annotated it with the different structural features that she saw. And they were able to see this transition between the intermediate and recognition complex, which was quite exciting. But something that is now available in version 1.0 and I guess 1.1 is a more automated approach for kind of encoding what has become the standard practice for analyzing these data sets, which is to really richly sample the model, generate 1,000 structures, and then do various clustering or maybe embedding analysis on that set of 1,000 volumes. And so there's various reasons why you would want to do this, but we've been able to apply these kind of landscape analysis methods to help interpret the results, identify rare states, and map interpretable reaction coordinates. In the last, how much time do I have? Am I out of time?
About
6 minutes. Okay. So I'll say something very briefly about revisiting ab initio reconstruction. So this was the overall architecture that I mentioned earlier, where we have this image encoder, volume decoder architecture. I left out how we go from how we get the pose, right? So the oriented coordinates, I magically assumed we already had that, but how do we get that? So there's a couple of possible paradigms for pose inference. Something that I think will be shared, or Fred will be talking about this later this week, is an amortized approach where you can train another model. So this is a very deep learning based approach where you just train a model to predict the pose given your input images. You can do this via gradient descent. So our model is fully differentiable with respect to the pose, so you can just differentiate your loss back into your pose variables. So that's this gradient descent based approach, or completely skip over pose estimation and just try to map the distribution. So that's this like GAN or GAN based approach. In our initial tests, these other kind of paradigms, this like variational or training another pose encoder model and pose gradient descent, ran into issues with spurious local minima. So this is this hand data set that I've been playing around with, and we're simulating kind of cryo-EM, so example images of noisy and noiseless images are given on the left, and we have these weird local minima with the amortized approach where we have symmetric states, and then pose gradient descent just completely fails. So we take kind of a hint from the rich literature in traditional reconstruction algorithms and do an explicit or exhaustive search. So given the current decoder, we just run a search algorithm over possible, the five-dimensional space of possible poses to get the best possible pose for that particular image. This has two features. The first is a hierarchical search procedure where we start with a relatively coarse grid and then successfully refine that, keeping the top K poses. You can choose K via branch and bound procedure, or you can just set it to be fixed. The second is frequency marching. So we start by band limiting our loss function to low frequency components. There's two benefits of this. One is computational efficiency, so we don't have to render the whole image. We can just render the low frequency components of the image. And this also prevents overfitting, because especially at the coarser resolutions of our search grid, there's no way we're going to be kind of aligning the high resolution details anyway. And so with the hierarchical search procedure, we can recover the correct hand cryo-EM structure. One very kind of additional feature of CryoDragon is that it's much more computationally expensive than the traditional approaches to render a given pixel. So in the traditional approaches to render a pixel, like some pixel in your image, you just take essentially the weighted average of the 8 spatially closest voxels. In CryoDragon, you have to go through the fully connected neural network. So this is like many orders of magnitude more compute to evaluate a given pixel. So CryoDragon models are much more expensive to evaluate. There were many hyperparameters to tweak, so this actually became a very fun engineering problem where like how do you exactly choose like all these different settings. There were some tricks to kind of split up the search between poses, like rotations and translations, which I will not go into. But after kind of basically pushing as much of the compute into image space instead of volume space as possible, we were able to really improve the pose estimation procedure to work on real data sets. So just very briefly, in CryoDragon 1, the original kind of publication, we attempted pose estimation, but it scaled poorly to real data sets. And with pose supervision, it's much faster. With CryoDragon 2, we can get reconstruction results in under 12 hours with the correct motions for the pre-Cullix spliceosome. I'll briefly mention cryofire. So this amortized approach to pose inference, which originally ran into the spurious local minima problems. Axel, Levy, and others in their cryo-EM paper developed kind of a new loss function that really avoids this local minima. So that was very exciting. I've been able to collaborate with them to bring that to the heterogeneous case. And I think the main goal here is to speed up pose search or pose inference, right? So we don't have to do this exhaustive search. And just showing a couple more results. Homogeneous reconstruction of this particular data set, various data. Homogeneous reconstruction with CryoDragon works well on a couple of different data sets. We learned a lot from this RAG1, RAG2 complex, which is shown here on the top row. And then we can also do a homogeneous reconstruction of the spliceosome with CryoDragon 2 in purple, matching the reference methods in gray. And again, here's the larger version, right, of that comparison, where the original approach took 38 hours, and there were various alignment artifacts. CryoDragon 2, we can reconstruct the correct motion, and it takes less than 12 hours. And just as a last example, this was, again, the RAG1, RAG2 complex, and it appears to be less noisy in the regions down here. And I think that has something to do with perhaps aligning on the correct symmetry copy, or basically not aligning so much to the relatively rigid core. So the goal is really to broaden the scope of cryo-EM to these complexes for which you can't get a consensus reconstruction. So just to summarize, I've talked about CryoDragon, which frames heterogeneous reconstruction as unsupervised learning of a continuous distribution of protein structures from cryo-EM image data. We propose this new neural representation for modeling cryo-EM structures, and the overall kind of deep generative model provides a very general, flexible framework for modeling heterogeneity. I think that's very relevant when you're thinking about which reconstruction algorithms you want to choose and what is the inductive bias that it induces when you're thinking about the distribution of structures that you get out. And with CryoDragon, we've been able to discover new structures and visualize continuous dynamics. And in the future, I'm very excited about the very nascent area and very much growing area of machine learning for protein structure determination. So a couple of different, I will go through this quickly, but I think there's a lot more to do in ab initio reconstruction, both on the engineering side, but also optimization and also identifiability of what are kind of variations in pose versus the actual heterogeneity in the structure. How do you characterize distributions of structures? So we have this kind of post hoc approach where we're analyzing the set of volumes, but I think there's like so much room to do actual benchmarks and atomic modeling. Ways of including other sources of information directly in the reconstruction process to either have better inductive biases for protein motion or just exploit all that we know about protein structure from these large-scale sequence and structure databases. I think this is a very exciting area that definitely has been changed because of all these other advances in machine learning for structural biology. And then in situ kind of CryoDragon and in situ subtomogram averaging approaches, I think is very exciting thing to kind of look forward to. Outlook in the post-AlphaFold era, I won't kind of go into this, but I think there's now this whole playground of tools and approaches to explore, and so this is definitely something I'm very much looking forward to moving forward. So with that, I'm happy to take any questions.